Good afternoon. I would like to welcome everyone who is joining us today for the Science of COVID-19 live webinar. My name is Agnieszka Carpenter, and I'm the Executive Director of BioME, also known as Bioscience Association of Maine. We're a trade industry group based in Portland, Maine, and we work with biotech and medical device companies and organizations in the state. We're very excited to host today's session and hear from the best experts in the state on the subject matter. With so much information about SARS-CoV-2 virus and COVID-19 disease in the media and the research findings changing every week, we thought it'd be a good idea to present you with information that's reliable and science-based. And therefore, we put together a group of amazing experts today. We will have 1 hour of discussion followed by 30 minutes of Q&A, and I would like to encourage our audience to type in your questions in the Q&A tab on the bottom of the screen, and around 4 o'clock we'll start asking some of these questions. I also wanted to thank all of our esteemed panelists today, Dr. Liu from Jackson Laboratory, Dr. Moore from Abbott, Dr. Mills from MaineHealth, and Dr. Maginnis from University of Maine, and also Dr. Douglas Sawyer, who will be moderating today's discussion. Thank you so much again. Today is a beautiful day, so we truly appreciate your time. And with that, I would like to introduce Dr. Douglas Sawyer from Maine Medical Research Institute.
Thank you, Agnieszka, and thanks to the Bioscience Association of Maine for putting us together. It's really an honor to be here and moderate this really great group of outstanding people from with expertise that spans the breadth that should be really making this a very exciting afternoon. So I'm just going to share a quick credentials on our colleagues, and then we'll get into some questions. Dr. Edison Liu is the CEO and president of Jackson Labs. Prior to that, he led the Genome Institute in Singapore. And I mention that only because during that time, he received the President's Public Service Medal for leading Singapore's scientific response to the 2003 SARS crisis, which is very, makes this very relevant today. We also have Dr. Melissa Maginnis, who's Assistant Professor of Microbiology at the University of Maine. She's an expert in virology and viral pathology and how our own biology interacts with virus pathology to determine outcome. So thanks for being here, Melissa. And we have Dr. Norman Moore, who's the Director of Scientific Affairs, Infectious Diseases in Abbott Labs. Dr. Moore comes to us as a expert in development of rapid tests for pathogens, having done that over his career many times. So welcome, Dr. Moore, and last but not least, Dr. Dora Anne Mills, who is currently the Chief Health Improvement Officer at MaineHealth, but in a former role served as the Director of Public Health overseeing Maine's CDC, and making her role especially relevant as well. So this is a great group. We have a great group of people online. It looks like something 250 or so people have joined us. I've got a number of questions that we'll go through, and then we'll open up to questions that you all are typing in, we hope. So I'm going to start it off with Dr. Liu. Wanted you to share with us what you think about how the SARS-CoV-2 is spread, and maybe talk about what we know, but also what we don't know.
SARS-CoV-2, which is the viral pathogen for the COVID-19 pandemic, is a coronavirus. It's the same family of virus that affected us in the SARS epidemic in 2003 and the MERS epidemic that raged through the Middle East. And the major mode of transmission for this virus is actually through respiratory aerosols. So when you cough, even when you speak, and especially when you try to project your voice, you generate droplets that go into the air, and it's primarily those droplets that will be transferring the virus. The other coronaviruses can be transferred by other bodily fluids, but the vast majority of the infectivity is really through these aerosols. The framework of the SARS-CoV-2 is that it probably arose from a bat coronavirus, but uniquely, it acquired very specific mutations in one of its coat proteins called a spike protein that made it exceptionally fitting to a human protein that allows the virus to then enter the cell. In fact, it's even more efficient than the earlier SARS virus. And I would say that that is one of the major reasons why it's so infectious, is because that fit just happened to be really superb, unfortunately.
So Ed, in comparing it to the earlier SARS-CoV, is that your explanation for why we're seeing such a different a pandemic versus a more localized?
Well, there's several other reasons. Besides the fact that there is a pre-symptomatic phase of shedding, which was much less likely in SARS. Individuals who were spreaders of SARS tended to be symptomatic, but we now know that in the COVID-19 pandemic, those infected, many are asymptomatic or pre-symptomatic, that is before they exhibit the symptoms. But indeed, every single piece of data that we know, from structural data to also experimental data, suggests that the SARS-CoV-2, the pathogenic virus for COVID-19 pandemic, is much more amenable to binding human cells than the SARS virus itself.
Thanks, Ed. Dr. Maginnis, I wonder if you could enlighten us on what your thoughts are. How do you compare this to the influenza virus that causes seasonal flu, and perhaps share what your thoughts are on why this virus is so much more deadly?
Sure. So there are a few reasons why SARS-CoV-2 is worse than the typical influenza. And the number one reason is that this is a novel virus. And just like Dr. Liu said, this likely emerged from bats, so we don't have any pre-existing immunity within the population. This virus just emerged in December of 2019, so it's only been circulating in the human population for the past few months. Influenza circulates annually, and we develop immunity from influenza virus, and we continue to build immunity because we're exposed to different strains of influenza every year. And so we can also have cross-protection immunity from influenza strains. Additionally, we have some vaccines for influenza, and so even if half of the population gets the flu shot, we have some protective immunity within the population, and then that reduces the number of flu cases. We can add that together with people that have the existing immunity that I just talked about, and then we have even more protection within the population. We also have treatments for influenza, like Tamiflu, people may have heard of that one. And we have medical personnel that have more experience in treating influenza, which is really helpful if people do become sick with influenza virus. And influenza has been researched extensively, and so although there are still many unanswered questions about influenza virus as well, all of that research is really useful in helping with the prevention and treatment of influenza virus. For SARS-CoV-2, we are living in a real-life experiment, and we're sort of learning as we go, and we need to learn all of this information really quickly and adapt really quickly as people are becoming sick at the same time. And so that's why I think we're seeing a higher death rate with COVID-19, because we're sort of learning on the fly.
Thank you, Melissa. Dr. Moore, would you like to add anything to that? And perhaps think about, can you help us think about the role of testing here versus we do some testing for the influenza, but we don't, not in the way we're trying to ramp up and get into testing in this virus.
There's quite a lot of differences. I mean, we learned a lot of lessons during that 2009 influenza pandemic when, you know, all of a sudden everybody hit, people didn't know what to do. We had a lot of people that were taking clinical samples, and I will say this, you know, at that particular time, the average was what's called the lateral flow test, where there are the little inexpensive cassettes, cards, and all that. And unfortunately, what we saw was that was novel enough. It was deeper in the nasopharyngeal passage, and if people weren't taking a good sample, some of the sensitivities plummeted down to like 15%. And so that's when a lot of people started switching to molecular and doing other testing. And this has been one of the unique things with coronaviruses. When it came out, we didn't even have virus to even make tests. Nobody even had attenuated or inactivated strains. So, you know, it has been a challenge for everybody to understand what these tests mean. And right now you can break that into two parts. We have tests looking for the virus itself. So, you know, like some of the molecular tests, and some of the ones looking for serology. And that's going to be further complicated by initially the FDA didn't look at a lot of the serology tests. And when they didn't look at the serology tests, they were put out with really poor sensitivity and specificity, and that has made a mess of everything. So we had a just rash of bad data come in. Now the FDA has kind of pulled back and said, okay, we're going to take a little bit more, but there's been a lot of complications along the way. Not having a virus to start with, having no good standard, and now we're even trying to understand what does even some of these tests mean. We're having cases of people that can produce virus for 30 or 40 days. So what do you do with a person that may no longer be infectious, but still may be positive by a test? And this is one of the unique things is there's a lot of questions still rather than answers.
Thanks, Norm. I wonder Ed, if you could comment on what your thoughts are on why certain patients and populations are more affected than others.
If you look at the epidemiology, the mortality, the death rates rise significantly with age. So if you're under 50, the mortality rate is the lowest. It's not zero for sure, but it's, you know, it's highly variable, but it's in a range of less than 1%, 1% or so. But if you're over 60, and for every decade, every 10 years over 60, the rate increases 10%, 20%, up to 40% death rate in some reports for people over 80 years of age. And so it's really heavily in that context that we know is a consistent basis. Now, in terms of different populations, whether they are ethnic or socioeconomic, much of that has to do with living conditions, whether or not they're crowded, if you have large families living together, whether people have access to medical care, and also the age distribution of the populations. Now, this has not been proven, but for example, the mortality rate that was reported in Italy compared to some other countries may be related to a certain degree to the fact that Italian demographics is actually older than in some other countries. But the challenge is that we're still, as all of the speakers mentioned, we're still learning a considerable amount. But there's some truths, and the truth is that as individuals grow older, the mortality and morbidity actually increases significantly.
Thanks, Ed. Dr. Mills, I wonder if I could just ask you to comment on whether that's what we're seeing here in Maine. Is that those same stats? You got to unmute yourself, Dora.
Sorry about that. So yes, it absolutely is. And as you may know, we are the oldest state in the country in two measures, both in terms of median age and the proportion of people 65 and older. So that's been a very big concern here in Maine. We are seeing that the vast majority of our deaths are amongst that older population, and about over half of our deaths in Maine have been from people who are living in long-term care facilities, either nursing homes or assisted living facilities. But we've also had, in hospitalizations, we have about 10% across the country and 10% here in Maine don't have any identifiable risk factors. And age seems to be by far the biggest risk factor. And then, as people may know, younger people under 65, it's things like hypertension that is not well controlled, diabetes, heart disease, severe asthma. Obesity is a major one, which we don't hear talked about too much, but it's really particularly people with a BMI, a body mass index of 40 and more. And we've seen people with those conditions who were younger than 65 pass away here in Maine and be hospitalized. And we've also seen, a number of older people. So it is panning out. What we're mostly looking at is not so much death rates, because we don't really know the death rate, but we're looking at death ratios. So the ratio of deaths over the people tested, the numbers of positive people tested, or positive tests, people positive who test positive in the population. And in Maine, it's around about 4%, which is about what we've seen around the country. You'd think, well, gee, we have an older population, maybe we'd be higher, but the tools that we've had to use, because we don't have, as Dr. Maginnis mentioned, the tools that we have for influenza, because we don't have effective treatment or vaccine, we don't have any kind of background immunity that we know of. So the tools have been, shutting down the economy and sending kids home from school and masking. I've got my Jill McGowan fashion mask here, and because I'm at work, and so as soon as I go out of my enclosed office, we mask. So we shut everything down, and that in March 15th, and that hey, good job, Doug. So I think that flattened our curve a great deal, and that has kept our death rates down. And of course, we also have a less dense population. We're not New York City, we're not Boston. It's really been mostly the major cities that have had by far the highest death rate so far. And we're not, we don't have it densely populated places here in Maine, but because we did shut everything down right when we were surging up the epidemic curve, in a surge, I think we got there, we did it just in time in Maine. So we've been able to keep our death ratios down.
Yeah, it's been actually nice to see that. It gives us a chance to think about the science behind this and how we can help the world navigate out of this. So I want to turn back to Melissa. You had mentioned that vaccines have helped us with the influenza virus. What's the process that we're going to get a vaccine for this virus, and how long do you think that's going to take?
Yeah, so the average length of time that it takes to develop a vaccine is usually about 10 to 15 years. And we're seeing that there are companies promising that a vaccine could possibly be produced within 1 to 1.5 years. So that's extremely impressive when we really think about that. And it's really a testament to the biomedical research community and biotech and pharma companies that are working around this pandemic. So there are currently 141 vaccines in development for COVID-19 when I last checked this morning. And those vaccines all go through preclinical development where we're trying to understand the virus infectious cycle. We have to understand the sequence or the genetic code of the virus. And then the researchers use that information to start developing vaccine candidates with the goal of producing immunity to the virus. So like Dr. Liu said, for SARS-CoV-2, when we see those images of the virus in the news, we oftentimes see the sphere with all these little projections that are coming off of the virus. And those little projections are called the spike protein. And there are lots of different platforms that are being developed for a vaccine, but ultimately we're trying to produce antibodies to the spike protein on the surface of the virus. And if we can develop antibodies to that spike protein, then we can help to prevent infection. So there are lots of different platforms to ultimately develop those antibodies. And when we're delivering the vaccine, it's like we're delivering the blueprints or decoys of the virus or parts of the virus into our cells and then asking our immune system to produce those antibodies. So we have to get through the preclinical development phase to develop those platforms. And then once we have made it through the preclinical development and we've tested them in animal studies, like in mice or in monkeys, and we determine that the vaccine leads to antibody production, then we move into the clinical phase of development. In the clinical development phase, we have clinical trials in humans. And we go through phase 1 through 3 clinical trials to test for safety and dosing and efficacy. And then once those initial tests are done, it starts to get expanded into different groups of patients. So right now we're testing in mostly the adult population 18 to 55, and that will be expanded into the older than 55 and the younger than 18 to also make sure that it works in the older population as well as in the younger populations. So there are a number of COVID-19 vaccines that have already passed through phase 1 and are moving into phase 2 clinical trials. And there's some really good early data that's being released from some companies, including the Moderna vaccine. It's showing production of neutralizing antibodies. So that's great news, but we still have to expand that into broader patient population and into larger groups. And then from that point, it then needs to be scaled up for manufacturing and distribution. We have to determine if we're going to need 1 dose or 2 doses. It's possible that we're probably going to need 2 doses of this vaccine. And the companies have promised some distribution by the end of 2020. And possibly we could see a vaccine by the end of this year, 2020, or possibly into 2021. And interestingly, the Advisory Committee on Immunization Practices are the ones that make the recommendations about who gets the vaccine first, because I know that everyone's sort of thinking about that. And it's going to go to frontline workers and healthcare workers first, and then to critical infrastructure individuals. So we may not see the vaccine available for the general population until 2021.
Thanks, Melissa. You know, even 2021 is optimistic, and I am reflecting on how we've yet to develop an effective vaccine for some viruses that we've been studying for a long time. What's your level of optimism that we will succeed with a vaccine to SARS-CoV-2?
Yeah, that's a great point. So when these companies are making these promises, that's all based on the idea that it's going to work. And so we don't yet know that it's going to work. But like I said, we do have 141 vaccines in development. And so I don't know that there's ever been such a force working on a problem. So everyone is gathered around this common problem and trying to come up with a solution. Some of the analysts have predicted that we need to have 10 to 15 vaccines in development to have a 90% probability of one of those working. And so I think it's great to know that if one of these vaccines that are moving along that are the front runners doesn't work, then we have a lot of others that are coming up through the pipeline, and there's a possibility that maybe one of those will work.
Thanks, Melissa. Norm, I want to turn back to you and get your thoughts on how we are going to be able to use or not use testing to get us all back together again so that we could have a non-virtual symposium on this someday.
That would be
back
good.
to work.
There's a lot of different debate. I actually heard one on like a TED Talk a while ago that was even suggesting a kind of a unique idea. You're 4 days in, 10 days off to try to eliminate that sort of ability when you're infectious, which was an interesting idea, leaving that aside, which I thought was interesting. Well, that's one of the big questions is how often do we test? Because this can get expensive too. So if you're, testing on that for, like maybe once a week, I think we all have to realize that, really there's not going to be any test that is ultimate sensitivity when you first start shedding virus. So, maybe sort of a once a week thing for some, obviously the ones that can do that well, the frontline workers. And of course, we still have to watch the basic things like symptoms. So, we can't get away from, that sort of thing. So that is the general hope. And again, I think we're going to try to see, believe it or not, a rush for less expensive diagnostics to come on hand, because this could get wildly expensive to have, a lot of these tests come out and just be testing people that much. So it's going to be multi-phase. And I think there's going to be a part for that antibody test. And I'm just going to say this. I don't know if we fully know what these antibody tests are going to mean. Everybody assumed at the beginning that, if you're antibody positive, you're done forever. You're the safest people around there. And there just was a story from one of the naval ships where they had been positive, they'd gotten over it, and now a few of the sailors tested positive again. We don't know if that's residual, a virus that they're shedding, but it did cause a little bit of concern that we don't know if there's potential for co-infection. So I think most of us are cautiously optimistic we won't be. But it's, again, as Dr. Fauci pointed out, it's not something we know either. So,
and I just ask you a question?
Yeah.
Can I say, so what do you think of the chances we're going to get some very good antigen tests soon? I know, I think Quiddix or Quiddle, there's a company that's got one that they're testing now. And because it just seems like with strep and even influenza to some degree, the quick antigen tests have been pretty successful. I'm just wondering what you thought the chances are that we could get one of those soon.
I think the chances are good. But the caveat is going to be, just like influenza, viruses rise, peak, and fall. So I think what's going to be happening with the antigen test is we're going to have a sweet spot where maybe they're going to be pretty good sensitivity maybe the day before symptoms, because viruses don't want you at home. It likes to have these nice high titers so that you go out and still spread it around. So they'll probably work pretty well 24 hours before, and maybe 3, 4, 5 days afterwards, clinical trials will tell, but probably drop off after that. So I think we're going to have a short window when they do work, and that's what we have to do is kind of put them in with other testing. But I wouldn't say that they're going to be sensitive for a longer range, but maybe that's going to be good enough.
Thanks, Norm. Hey, Ed, what are you guys doing at Jackson about using testing to get people back to work, and how so?
Well, in March, we made a decision that we would, because just my personal experience is that testing is the core to how you control a pandemic, and I was not very sanguine about the efforts that were going around. So we decided to stand up our own test, and as I mentioned to you before we started, we now are doing 1,000 tests a day, and we've done our 16,000th test as of today, and we will be doing 20,000 tests a day starting in July, in the mid-July. And that is really to support both our home states of Maine and Connecticut. And right now, Connecticut has a much bigger problem, as Dr. Mills mentioned, than Maine is. But the key is actually to have it ready. So in that context, let me just say how we made a decision to do universal testing on all our employees, which is 2,400 in 4 sites in 3 states, is one thing is perfection is the enemy of the good, and everybody's going to have an opinion, right, Dora? Right, Dora? You being head of CDC, you know exactly what I mean, and so we just have to do what we think is correct now, and we need to learn from that. And so we started to test all our employees. Let me just say, when this first started out, half of our employees were more or less sent home to do work from home. The other half needed to be around because we are considered an essential sector. And we did all the necessaries, right? But to bring people back as we're seeing activity returning, especially in the research end, because we're actively involved in the research in the COVID-19 issues, we have to bring people back. And so the thing we decided to do was to test everybody, and in an orderly fashion, doing it in such a way that we're learning on a consistent basis. We're doing a retesting every 2 weeks for 2 cycles, simply to understand what the conversion rate is. Because everybody talks about, as Norm was saying, you know, you got to do it twice a week, or you got to do it once a week. We really don't know. And in the context of Maine, where the frequency in the community is actually low, that may be considerably overkill. But there's another reason for that, and that is to understand, you know, that human beings who work together, they have a psychology, they have fears, they have concerns, and if nothing else, if we can say to them that when you come back to work, it would be COVID-free as much as possible. And if we could do that, then it will help at least the psychological comfort of our workforce.
Thanks, Ed. How has that been received by everybody? Is there anybody that has balked at that?
Well, right now, I would say we've had remarkable support from our workforce. There's always concerns, and the key for us is never to dismiss a concern. So there was a tremendous amount of prior work to discuss with our employees, to have groups where we had conversations, explanations from middle management all the way up to my intervention as well. And we were not doing it all at once. We're doing it in sectors so that we would learn from each sector what are the concerns, et cetera, including the issue of comfort in terms of sampling, as Norm will know, and other aspects like that. So we keep them informed very much along the way, very much like Nirav Shah has done, I think, a wonderful job in informing the general public in Maine. And I would argue that's one of the reasons why you're not having massive panic in this state, is because there's been a lot of communication.
Yeah, and I think you're right about that. Dora, do you want to comment on how MaineHealth's approaching the return to work and whether testing will be part of that?
Yes, well, we are actually, as Doug knows, we have an app that we have on we're screening everybody as they come into our facility. So you have to answer questions every morning, and you get a badge. So this is mine on my phone, which clears me till 3 a.m. tomorrow. I hope not to be here at 3 a.m. But so this, you have to answer if you have any questions, if you've traveled. So you have to attest. And a lot of health systems are using that as a screening tool when you come to work. And And then, of course, we have a lot of people in administration who are in IT and the like who are actually working from home right now. But as we're starting to bring them back in, we are screening them that way. And though we also realize that, because increasingly last night there was a study out of JAMA, published in JAMA, showing, and every week there's another study showing how many people are asymptomatic. So, and particularly the younger you are, it appears that if you test positive and you don't have symptoms and you're older, as we've seen this in a bunch of nursing homes here in Maine as well as around the country, that you're likely that you're pre-symptomatic and you're going to have symptoms. But then if you're younger, this new study shows about 42%. It's a small sample, but 42% never had symptoms, and it was mostly younger people. And we're seeing that in the epidemiology as well as some of the studies. And we just know that younger people, our kids and young adults, aren't getting the symptoms as much as anybody else. They're not getting severe illness either. So there's screening. I agree, screening and testing are sort of you've got to have that as a foundation, but we're still in the baby stages, as Melissa mentioned a while ago, that, it's only been around for 4 or 5 months or so. So we're still learning about this and so we're not testing. We have almost 23,000 employees. So we really can't be testing everybody right now, but we are having them attest to symptoms. And then we also, if they come back to work and they obviously start having symptoms, we get them tested. So that's been our major strategy. It depends on your numbers, and how, so and I think particularly, Ed, you have a lot of staff, employees in Connecticut, which has been one of the hardest hit areas in the country. So it makes some sense that you would want to screen them and test them as well when they come back. We haven't been hit nearly as hard, and with 23,000 employees, it's been almost impossible to actually get a test on people when they, for everybody. But so that's how we've been. I think for different places, different situations, you have a different type of screening and testing strategy. I just been talking to a couple of colleges here in Maine, Bowdoin and Bates, for instance, who were thinking about they're going to be bringing people from all over the country, their students, and hopefully the world if the borders open up. So it makes sense then for them to be testing everybody when they get on campus. Get here as nonstop as you can, and so you don't pick anything up as you travel, and then get tested. So they are looking at doing some tests when people start coming back on campus, and that kind of makes more sense for that situation.
Thanks, Dora.
Can I just add to
Yep.
Dora's comments about the universities? We certainly have contacts in China, and what they're doing in some of their universities is every single person has to be tested and they're cleared, just like what Dr. Mills has shown. But because many of the campuses who are not urban are actually out in not quite the countryside, but the suburban areas with dorms, they stay there for a period of time until they get retested again. So it's the old cordon sanitaire, that the old infectious disease people way back 200 years ago did. You make sure that the people within a cordon don't have the disease, and you don't let people come in and out. Yeah.
Thanks for sharing those. Melissa, could you share with us as a virologist what you think are going to be the treatments that we're going to have, what's in the works, what's being talked about on the airways, and what do you think is the future going to look like?
Yeah, so currently there are no FDA-approved drugs for the treatment of COVID-19, but there are over 200 drugs in development, and that's the ones that we know about, so there probably are more than that. And in some cases, there are investigational new drugs for COVID-19 that can be issued an emergency use authorization, such as remdesivir, which people may have heard of, and that can be used for critically ill patients. And this drug was offered FDA emergency use authorization for patients with severe disease. And what it does essentially is it acts as a blockade in the replication of the virus. And so when the virus infects a cell, it essentially lays down its blueprints, its genetic material, and the blueprints get copied. So it's like going through a copy machine, and then there's a paper jam once remdesivir is introduced into the cell. So the virus can no longer copy its genetic material, it can no longer replicate, and it can't produce new viruses that will then escape from the cell and go on to infect new tissues and new hosts. And so that's a very effective type of antiviral drug. So we may see other drugs that are like remdesivir. So these are nucleoside or nucleotide inhibitors. And we may also see drugs that would block different steps in the virus infectious cycle. So I mentioned the spike protein. That protein binds to a receptor on the surface of the cell, and it's ACE2. That was the combination that Dr. Liu was talking about. So spike has to find the ACE2 receptor. Once it finds that receptor on the surface of the cell, then it sort of unlocks the cell. So if you can block that interaction from happening, then you could block the initiation of infection. So we have to understand all of the basics about the infectious cycle of the virus. And when we understand them, then we can think about designing inhibitors that can block all of those different steps in the virus infectious cycle. There's another treatment that's also being used, and that's convalescent plasma. People may have heard of this too. And this is through FDA registered donations from people that have recovered from COVID-19. And then they can offer this donation, and their antibodies can be used and administered to people who are critically ill with COVID-19. And those antibodies actually can lead to recovery in those patients. And so those are the only two treatments that we currently have for COVID-19, but I think that we'll continue to see others coming online. The process for going through all of the approvals is similar to a vaccine, although it's not quite as cumbersome. And that's because when you're using an antiviral treatment, the person's already sick. And when you are introducing a vaccine into someone, you're introducing it into a healthy population to develop immunity. So there are cases where if someone is ill with COVID-19, it presents this opportunity that you can offer emergency use authorization or compassionate use where there's no other hope for that patient, and then you can try these investigational new drugs to see if they could actually lead to some recovery in those patients.
Thanks, Melissa. Melissa, there's a lot of talk about the repurposing of anti-malarial drugs in the press and whatnot, and there are clinical trials that have been going on around that. Can you comment as a virologist what your thoughts are on that?
Well, I think that we've seen really some mixed at best results with the use of things like hydroxychloroquine. And at this point, the FDA has urged against the use of hydroxychloroquine because of those mixed results. And in some cases, those results have had significant effects on the health of the patient. So I think at this time, those studies are probably halted for good reason.
And is there, one of the folks has asked a question about whether the adverse effects of those drugs could be due to their effect on the immune system, in particular interferon gamma. Do you have any knowledge on that front?
The effects of any type of antiviral drug?
No, hydroxychloroquine or chloroquine on suppressing interferon gamma and that leading to adverse consequences in this context.
I'm not sure. Maybe someone else can comment on that.
The most prevalent side effect that was raising a lot of concerns in several studies is cardiac arrhythmias. So some of that has been known before, but I wouldn't be surprised if part of that is because people are hypoxic or they really have, lung problems, significant lung problems, which always affects the heart, and you compound effects to compound side effects.
And of course, there are some folks affected severely enough to have direct, or what appear to be direct cardiac effects of COVID-19. So let's switch gear to a little epidemiology lesson. So Dora, why is flattening the curve so important for the hospitals? And then could you comment in this answer on how PPE and masking plays a role in that or not?
Huge. So yeah, so it's probably, it was kind of funny to hear everybody, it seems like around the world and the country, understand what flattening the curve was back in February and March and April. So, I'm so used to having to explain it for so much, and all of a sudden everybody kind of knew that, so basically, you're trying to prevent this big surge that can overwhelm the healthcare system, the hospitals, as we saw happen in New York City, and it's happened in other cities around the world, and countries like Italy, so where the hospitals just became completely overwhelmed and the economy shuts down because of the disease itself. It just making so many people sick and everything just overwhelmed. So people really wanted to avoid that. And I'll just back up and tell a little bit of story. So in Maine, we got the test kits at the state on March 9th, which is a Monday, and March 10th at Nordics Labs here in our MaineHealth lab, we were able to do PCR testing as well. So we started, we got our first test result positive on the Thursday the 12th, and it was somebody who traveled to Italy and was quarantined, so it didn't seem very worrisome. And then Friday and Saturday and Sunday, we got a bunch more, several more, I'd say about a dozen or so were positive, mostly in Cumberland County, a little bit in York County. And when the state was doing the epidemiology investigation and contacting them and finding out, did you travel? Did you know anybody with COVID? And some of them had come back from Italy, whatever, because February vacation had been just a couple weeks earlier. But a number of them did not know anybody who'd had it. They hadn't traveled. Interestingly, several had traveled to New York, which we didn't know was having it too much at the time. But it was very concerning because what you could see very quickly was that it just wasn't one or two. We had about a dozen quickly within a couple of days who had no idea how they got sick. And what we knew is that we had community transmission here in southern Maine. And we also knew that because we were, the state and everybody was saying, you can get tested now if you have symptoms, if you were in a high risk category. And we were every day, the number of positives was doubling. And so on Sunday, the 15th, it was when the governor said, shutting the state down. We got to shut everything down because we were on that curve. We were on that curve we needed to flatten. And it was a curve, our doubling time, that is a number of days it takes to double the number of positive tests, was a day. And we knew New York City was a couple of days, it started becoming a couple of days by then. And so we knew we were going up, it's an exponential growth. And it's very frightening to see. And we didn't have a vaccine, no treatment. All we had was to say, everybody stay home, shutting schools down. I remember the day very well because it was March 15th. And for those of you who are historians, know that was a major day in history for two reasons. It was the Ides of March, the day that Caesar was murdered, and it was also the bicentennial from Maine. So we had a lot of celebrations planned that all got canceled. And so the state shut down. And honestly, the next 2 weeks was probably the most scared I've ever been in public health, because we did not know if this was going to work. No country had done this. The United States had never done it. The United States had never done it. We had never done this, and it's statewide. Even the 1918 pandemic, they did it in some cities. We had never done it statewide and across the country. We didn't know if it was going to work, but it was the only tools we had. So the doubling time continued to be 1 to 2 days for the next 2 weeks. It was very scary. And we started filling up Maine Medical Center, Mid-Coast Hospital, Southern Maine Medical Center, all our big hospitals started seeing patients. It was very frightening. And then, you knew it was going to take a couple weeks. And right around April 1st, it started flattening out. And I didn't say anything. We have every day we have a incident command system call, and I didn't dare to say. And then about 6 days into it, about April 6th or 7th, I said, "Oh my gosh, our doubling time has gone way up." That's the one thing you want to go up. And the numbers of cases, the hospitalizations went up we're still going up because that's a lagger. You know, your cases go up, and then as people get sick, it's about a week into illness, they usually get hospitalized, and then your deaths are about a week or two after that. So our hospitalizations were still going up. But I started telling everybody here, I said, you know what? I don't want to say this publicly, but we're flattening the curve, I think. And so about April 12th or so, I was just in tears. It was so exciting to see. Now, it was horrendous to look out. I'm downtown Portland. You couldn't see a car anywhere. You could walk down Congress Street and you wouldn't get run over. So you knew these businesses are closed. I mean, it was exciting to see from the epi standpoint, but we knew the cost was also to our economy, but it was the only choice you have. The pandemic itself will close your economy if you don't flatten the curve. So the economy gets closed down no matter what. But to see that our hospitalization and eventually our hospitalizations peaked around the 8th of April, and then they started going down. So it was amazing to see the curve flatten. And it happened because people were staying home, they isolated. It was just astounding to see. So now, I just quickly mentioned, we've got these tools still we have to use because we have finished chapter 1. This is a long book. This pandemic is not going to go away anytime soon. So we've got got my mask. We've got to do social distancing, stay 6 feet apart from anybody who's not in your household, and got the Split Rock Distillery has made, we contracted with them to pivot and make us hand sanitizer at MaineHealth. So every one of us has this with us, and I've got one in my pocket, so a smaller one in my pocket. We've got to do hand hygiene, masking, and social distancing. Those are the tools we have right now. We don't have good treatment or vaccine, and this has not gone away. So we're in chapter 1 was flattening the curve. Chapter 2 we're in right now, which is coexisting with COVID. But we can't it's going to overwhelm us again, or it will overwhelm us. It didn't really overwhelm us, but it will overwhelm us if we don't pay attention to all these 3 major tools.
Thanks, Dora. Thanks for sharing that experience at MaineHealth. Norm, when we get to chapter 5 or 6, is it going to be necessary to do sentinel surveillance, like, random testing, like pick a letter of the alphabet, everybody with that last name that begins with that letter gets tested one day of the month to know where, things are happening? What are your thoughts on that? Norm, are you frozen? Okay, how about, what do you think, Melissa?
I think that testing is really key to ensuring that we don't have additional outbreaks. And that's because people can be asymptomatic for 14 days. And during that time, they can be transmitting the virus. And so if they're out transmitting the virus, and therefore they could be spreading it to everyone that they're interacting with, and we don't have any read on that. So I think it's really important to ensure that we're testing. And then from there, we have to follow the protocol of test, trace, isolate. And so once someone does become positive through a test, then all of their contacts need to be traced, and that individual needs to be isolated. And so we need to ensure that so that we don't have, again, like a rising of the curve, like Dr. Mills was just talking about. What I would like to see, what I hope to see, is an increase in the testing capacity so that we can have some type of sentinel testing that's going on, hopefully throughout the state, and especially in larger organizations and organizations where people have to be in close contact, like on college campuses and things like that, so that we can ensure the safety of all the people within our own communities. I think that will be really central. I think that Norm maybe is
Norm's back.
Unfrozen, so if he wants to add to that, he can certainly do that.
For some reason, my internet seems to have a little problem around this time of the day, so hopefully I'll be back for a little while. So I didn't hear quite the question, but I'm going to add, there's going to be a chapter 3 too.
Yeah, okay. So just thinking about sentinel surveillance and whether we need some broad testing strategy that's not symptom-based, but more, or workplace-based, but more broad. And would that be part of chapter 5 or 6?
Well, let me throw one other thing that may be happening is we actually don't know what's going to happen. I think at first we didn't know what was going to happen in the summer, and we didn't know if it was going to be like 2009 where it's going to rage right through the summer, or like the 2000 or the 1918 where it kind of took a little lull and went back up. this being unique, where we've had, I think we're expecting a bump up as we open up the state. But my anxiety is actually going to come in what happens when flu and COVID are co-circulating, because I don't think we feel that they can be mutually exclusive. And are they going to have additional morbidity and mortality when they're together, depending on hopefully we have a strong vaccine with the flu. And I'm going to beg and plead every person listening to tell everybody else that you must have the flu vaccine, because I can't imagine a more important year that everybody should have that flu vaccine, because I don't know what's going to happen. And I know I'm skirting the question, but I just want to kind of get this point out there. This is going to be a unique winner if they co-circulate.
Great point. Great point.
Let me, people have, you don't need to make it mandatory. I mean, Iceland provided free testing for any volunteer, and they got almost 9,000 people. That's enough to sample a population, certainly their population, which is relatively small. And with that, you can actually get secular trends, but I do think that two things. One, you have to test vulnerable populations before it breaks out. And nursing homes is really the key thing. I know Governor Mills and her administration is really pushing hard on that. I can tell you, Governor Lamont in Connecticut, where we're actually deeply involved in some of their planning, they've actually mobilized the National Guard so that they can cover all the nursing homes for testing. Okay, so that's important. But let me just say one very important issue here is the rise that Dr. Mills talked about when we were watching was actually the rise of hospitalizations of sick people who were then tested and were COVID positive. That's, quite frankly, the tip of the iceberg in a population. By the time you get a rise in hospitalized people who are COVID positive, the pandemic, your epidemic is already raging in your population. So that's why population-based testing is so important. You want to get ahead of the curve before it hits. You have to isolate before you have people already present in intensive care units.
So you're advocating for some volunteer population-based testing as done elsewhere. I like that idea. What do others think? Dora, what do you think of that idea? You're muted.
Yep, thank you. Sorry. I think we have been in a lot of discussions, as Ed and others know, about this. I think you have to look at both the prevalence of disease, which we don't really know, but we have some idea in terms of test positivity. So of those who are tested, what percentage are positive? Right now in Maine, it's about 4%. So we need to be a little bit careful. I think right now, if we're going to, it depends on the purpose of testing, but if we're going to use testing to try to detect if there's disease or not, you do with a low prevalence of disease, you can have a very, a positive test can often be a false positive test, right? So you have to be a bit careful. But I think in southern Maine, we have a bit higher prevalence, and we've got places where people are at high risk for severe disease, such as long-term care. I'm worried right now, particularly around the assisted living facilities. There was one, I can talk about this because it was in the news, but Cape Memory Care, which is assisted living facility here in southern Maine, we had 10 admissions over the weekend from there. And assisted living is different from nursing homes, and the nursing homes have nurses on there. You've got healthcare providers. Assisted living and group homes, people are mobile. It's almost impossible to do social distancing and masking, particularly if they've got intellectual disabilities, or this was a memory care unit, so they're not going to keep the mask on. So it's very difficult to minimize the disease transmission, and there's no healthcare there on site. So I think testing in those places is also perhaps warranted. One thing we're looking at with the nursing homes is actually testing the staff rather than necessarily the patients. And because it is actually, nursing homes right now are closed units. You cannot go visit anybody in a nursing home. So those outbreaks in nursing homes are actually a symptom of disease out in the community. It's the staff who are, that many times don't know they have it. They are asymptomatic, they're coming to work, and but because you can't do the social distancing, you can't put masks on everybody, stay apart, then they're spreading it. It can convey it within the nursing home. So, so one strategy might be to test the staff in the nursing homes, as an example. So, so that's all being worked out. And then you've got colleges and universities, I mentioned earlier, people coming in from all over, places with different, much higher prevalence very often of disease, or we think they have. So then testing them when upon arrival. So I think you have to look at what the purpose of testing in different situations is, and each, each situation is very different. And but anyway, there's a couple of other strategies that we've been contemplating with Ed and others.
Thanks. We're going to open up to the questions that are being written in a couple minutes, but we had one question that came in before the panel got together. I'm going to read it to you because it's interesting, and I'm going to editorialize slightly on it because so my question to all our panelists is, why Maine? With all the resources and different workforce traits in other parts of the country and even the world, how is it that our state is punching so far above its weight? And this is where I'm editorializing, with all due respect to the heavyweights on this call, when it comes to success in biotech, and particularly during COVID-19. We saw Abbott first come out with their test for the novel coronavirus, and now IDEXX has one too. The speed with which these were developed is hard to fathom. How did this happen in the great state of Maine? Perhaps more importantly, how do we ensure it continues to happen here? So I wonder what you guys' thoughts are. We got the world leader in mice models of disease on the phone here. We've got chief scientific officer at a biotech company. We've got a world leader in biology at one of our great universities. What are your thoughts about this?
If you want me to start, I will actually say there was, in the earliest days of diagnostics, there was actually this little division in, believe it or not, Corning, the people that made glass, they had a little diagnostic division. And surprisingly, there was a company early in Maine that was called Bentrex. And amazingly, David Shaw started there, Roger Piazza started there. So David Shaw went off to form IDEXX, and Roger Piazza formed BioME, and apparently in the early days you had to have an X in the name to start biotech in Maine. So it was just amazing that one company really spawned off some really brilliant stuff. I mean, you look at IDEXX; they are a phenomenal leader in veterinary worldwide. So you look at, I was amazing that one little hub really spurned off a lot of people that were able to. You don't think of Maine as a biotech hub, but I think that really kind of did start a lot of that, at least on the biotech front.
Thanks, Norm. Ed, what are your thoughts?
Well, I've always been bullish on Maine. I mean, Doug, I've talked a lot about it. For what it's worth, the smallness is our advantage. The fact that we're close to the leadership and can talk to them in a direct manner, I think is really an advantage, and to have the connectivity. But let me put it this way: there's also, I think, people have different opinions, that, and so forth. We're not any different. But I think on the whole, people tend to want to work together more here than in other states, and I do believe that catching the curve before it really turned upward was a key issue of why we're still not having the same problem as New York or Boston and what have you. The problem is that can't go on forever. As you know, our economy is so dependent on tourism and everything else. And my hope is that we can use the same spirit to actually thread the needle. Going forward, the challenge is that what I think several of my colleagues here mentioned, we have to learn to live with SARS-CoV-2. I mean, just like we've learned to live with influenza and every other infectious disease, we're just going to have to learn to live with this. That includes making sure we have the vaccines, et cetera. But I also think that some of the operational tactics that you've heard here are precisely the things that are be temporizing before we have those treatment. My only request is that we learn from everybody's experiences. you know, you might not agree with our approach to test every single employee, but I can tell you we're going to learn from that, and you know, if it doesn't work, it will change.
Yeah, I think it's interesting that you know, we've got some real innovative people who are science-minded and are doing things like you're doing, Ed, or Dora's doing at MaineHealth, or Norm's doing, that are really going to contribute to a global knowledge of how we're going to live with this. Melissa, what's your take on the answer to how Maine is punching above its weight?
Yeah, so I just wanted to add that I think Mainers are so resilient and such flexible thinkers, and we've seen that resilience in so many different ways. In one way, we've seen the University of Maine students who were all sent home before spring break and asked to continue their education online. And we've seen that in our schools, in our K through 12 programs as well. Our students are now being homeschooled. And it's really amazing to see that at the University of Maine system, our retention rate was actually higher this year than it was in comparison to last year. So why is that? I think that people are rising to the challenge. And I think that people in Maine, and especially our students, they're ready to roll their sleeves up and get to work. And I think that with our excellent education system, we're able to train our students to go out and work in these companies where they can serve not only to really help to contribute to things like research and the development of diagnostics, but also to be leaders. And so I find that to be really inspiring and really encouraging. And we've seen lots of great partnerships. So the University of Maine partnered with many different companies. So they've partnered with places like Maine Medical Center, and also with local distilleries who flexed and started making hand sanitizer when there was a shortage of hand sanitizer. We saw our nursing students actually have an accelerated time to degree completion so that they could get out to work and start to actually work on the front lines. So we've seen so many wonderful things, and I think it really has to do with the resiliency of the people in the state of Maine, and I think it's wonderful.
Thanks, Melissa. So I'm going to turn to the Q&A, and I'm going to select, because we've got over 65 questions in here. I've tried to address some of them. I think we have addressed some of them along the way. But I want to start with the summer season is upon us. And there are a couple questions that relate to that. One is to do with our visitor population that comes in the summer to enjoy our great state. And the other is has to do with kids and what their role is in going back to camp or school. And how do we think about that from an impact on the pandemic locally? Dora, you want to start with?
Sure. I'm actually on an expert panel of 6 who have been coming up with the checklists and the guidance for the state, infectious disease and public health experts. So I've been immersed. We've been meeting about 2 or 3 hours a day for, I think, 5 weeks now, most days of the week anyway. So we actually worked very hard on the summer camp. As an example, restaurants, tattoo parlors. I mean, we've looked at all kinds of things. We were doing harness racing the other day. So I've learned a lot about sectors that didn't necessarily know a lot about before. But in terms of the tourists coming back, there's several really sticky issues. I mean, one with tourists, right now they're coming from areas of much higher prevalence. You just go across the Kittery-Portsmouth Bridge. Southern New Hampshire is a big hotspot for COVID. Massachusetts is one of the hardest hit areas in the country. They've had almost 7,000 deaths. They might have gone over that today, which is much more. I think it's about almost twice as much as what the whole state of California has had. California is about 38, 39 million. Massachusetts has 5.8 million. Massachusetts has had more deaths than the whole country of Canada. So we've got hotspots. Connecticut, New York, everybody knows has been hot. So we're, viruses, pandemic viruses particularly love to hitchhike on travelers. So we've got to be very careful. And the issue particularly is those travelers who are coming in and staying at lodging facilities, because they tend to have to they're not staying in the hotel most of the time. They're going out, they have to eat out 3 times a day. They're going out and about in the community. So if somebody's coming up, frankly, to their summer cottage, their seasonal cottage that they own somewhere, I don't get as concerned, but the highest risk people are really, I think, the people who are going to a lot of our tourist spots and staying in hotels. So they're a number of people who've been trying to work on figuring out how can we get them up here in a safe way so we can open up our economy, which is so dependent on tourism. And we're floating some ideas. I don't want to talk about it publicly because I think I just realized there may be some press on here. But there are ideas that are very much under discussion for July 1. And of course, hopefully the prevalence will go down in those areas as well between now and July 1. And then the summer camps, you know, they're more quarantined. I mean, they're coming in, those kids are coming in, and you can look at summer camps as being kind of a cohort, a quarantine group. I was the state health officer, head of Maine CDC during H1N1, the pandemic from 11 years ago. And we had a lot of summer camps that came in and had a lot of outbreaks, but it was okay. They did fine. The camp directors knew how to cohort the students, get them, screen them, get them isolated. And so it worked well. So we're using those same principles of isolating them, cohorting them. They're already cohorted. They're usually down a dirt road somewhere, so they're not interacting with the public. And we're asking them, actually, the camps, the summer camps, not to send their kids into town. And the staff, once they get there, their days off, they're staying on campus. They're not allowed to go into town. So we're kind of looking at them as a quarantine group. And then, of course, colleges and universities, farther down the line, as I mentioned earlier, we've been working on those, and K through 12. But some of the same principles apply of you know, trying to screen and make sure that students and the faculty, whether it's a college or K through 12, you know the symptoms, be able to stay home, and then cohorting them as well. You can't do that on every campus, but a lot of elementary schools, as an example, have 3 wings. So each wing is a cohort. We're doing that in nursing homes. You have a cohort of, say, a wing that students and faculty, they are there all day, and then they don't interact with the other wings. So you don't have faculty who go back and forth or staff who go back and forth. You keep them as a cohort. And that way, if you have an outbreak, it's limited, hopefully, to that one wing and not the rest. We've had Maine Veterans Home has 2 huge wings. One wing had a major outbreak, but because they cohorted and kept the wings separate before the outbreak, the other wing was untouched. so those are some, and then of course the masking and the social distancing and the hygiene, the hand hygiene. So those are all the strategies we're looking at, but hopefully, again, as part of the coexisting with COVID, and how do we open up and go back to functioning, but also coexist and not let it surge. We want to keep that curve as flat as we can.
So should we hand these out at the border as people come back to Maine on vacation?
Yeah.
Change topic. All right. So Norm, you're a few questions come in here about testing wastewater, which I guess is being done in other parts of the world. What are your thoughts on that, about a way to look for communal spread?
I have not seen testing wastewater, but at least with the CDC, they've come forward and said that we don't expect a lot of the services to really be as strong a way to, I mean, I'm not saying that they can't transmit, but I have not been seeing anybody really worrying about the wastewater. So that's really been, I've seen other pathogens that way, but we're not talking about Giardia and things like that. I've not, I mean, I don't know if anybody else can answer that, but wastewater goes into a treatment plant, and these are envelope viruses. They're not the hardiest, they're not spore formers, not the hardiest things. So I'm not expecting, without any data in front of me, that they're going to last long in wastewater. But if anybody else wants to comment on that.
Melissa, you had your hand up
Yeah,
there.
so I'll comment on that. They are doing this in other parts of the world. It's been done in Paris, and we're starting to explore that possibility for University of Maine campus as well, as a way just to monitor what could be going on within the community. And it has been shown actually that the virus can be shed in the feces, and that is infectious virus. So it's plaque forming unit virus, so virus that's able to infect cells and destroy those cells. So not just RNA coming from the virus. And so if infectious virus is being shed in the feces, then We do need to be especially careful in the bathroom settings as well. So that goes together with hand hygiene. There are some recommendations about, closing the toilet prior to flushing the toilet to prevent the spray and the aerosolization of the virus. So those are some things, some new research is coming out and actually showing that virus can infect the intestinal tissue. And the intestinal tissue does have the ACE2 receptor that the spike protein binds to. And so that's likely how it's getting into those tissues. So there is definitely a possibility that there's infection going on in the GI tract. And that is one of the symptoms too. One of the early symptoms that people have noted were some GI symptoms. And so if people are having GI symptoms, it's probably not something to ignore and just think that, oh, maybe it's just a stomach bug. That might be one of the symptoms where you say, well, I'm not sure, but maybe I should just stay home to be safe, because it's probably better to be safe than sorry. But that's new research is coming online about that. And I think it's something that we all should remain aware of and start to possibly consider the possibility of looking into wastewater testing as a way just to monitor in our own communities what could be going on.
Thanks, Melissa. Related questions that are coming through are asking about how long the virus survives under different conditions. So, for example, the wastewater is one, but what about in a setting that's exposed to sunlight or UV light? Does that matter? Could you comment on that while you're on?
Sure. So there have been some studies about how long the virus can remain stable on various surfaces, and it can remain stable up to 3 days. So there is a possibility that if you touch a contaminated surface, like the top of your desk or something in a store, that there could be infectious virus there, and then that's how the virus could be transmitted. So that's something to be aware of. We know that the virus is also spread through respiratory droplets and through aerosols, and it can remain within the air. The length of time can vary, and that kind of depends on various conditions like the temperature and humidity and things like that. But it can remain in the air. I've seen up to 14 minutes. So that's a possibility. So if someone is to sneeze or cough and you're in the same vicinity as that person, then you could actually inhale the virus from their respiratory droplet or aerosol transmission. Also, if you were to walk into that space after that person has left. And so that's why it's really important to ensure that we're wearing the face covering so that we can prevent those types of things from happening. And the face coverings really is about preventing, if you are infected, preventing the transmission to others, not so much protecting yourself. And so this is a time in our lives where we actually can protect other people. There's very few times in our life where we can say that we can protect our fellow citizens within our community. And so that's why we're wearing face coverings. In terms of treatment with UV light and things like that, there's research showing that UV light actually can destroy viruses. However, it really depends on the type of radiation, and it also depends on the strength of the radiation. So as a UV bulb actually starts to die off, it's not quite as strong, and it might not actually deactivate the virus. So we do need to be really careful with those types of treatments. Thanks,
Melissa. We got a couple questions here from Representative Chris Babbage of Kennebunk, and I want Dora to take this one, I guess, to start. So a couple questions from Chris's constituents about children. What's the, you want to take a look at that? But basically, what's the danger of reopening schools if children are not targeted like seniors are by this virus?
Yes, and hi, Chris. I go way back with Chris. So, yeah, so that we didn't know, when the schools were shut down in the middle of March, really wasn't known if they are major transmitters. We know with influenza, they are major community transmitters. In fact, you can make an argument that if you've got a bad influenza season or any influenza season and you have a limited amount of vaccine, even if the seniors are disproportionately impacted, you might want to vaccinate all your school kids first, because they're the big transmitters. They're in a petri dish all day long, and then they go home, spread out around 25 households across the community and spread it. It's not known exactly if they're major transmitters of COVID. We know that they're not impacted. They're not at risk for severe disease. Some kids do get severe disease, but it's rare. So, but we're learning more and more that they may be contracted, and they're simply not symptomatic most of the time. It also may be that there's antibody crossover with protection crossover with other coronaviruses that cause common colds, which they tend to contract more often. So it's not really known, but I'd say that I think everybody wants to open up schools in the fall for lots of reasons. And obviously, if we're going to learn a lot more, the good news is that there are schools in Asia and Europe that are opening up now, so we're going to learn from them. And there are year-round schools that are opening up in California and other states. So we're going to have a lot more information by August. But I think one thing that pandemics always do is they always shed light on the cracks in society. And one of the major cracks we've seen we've shed light on, and more light that a lot of us know about, but that shed more light on, is health disparities. And so we've seen our frontline service workers who are low income be disproportionately impacted by this. They're contracting this infection. People bagging groceries and working in food processing plants and serving people in long-term care facilities and group homes. Likewise, with the schools being shut down, we know there were a number of students who are at home that school districts have not been able to be in touch with. The child abuse across the country, the child abuse reporting is way down. People aren't seeing these kids, so their child abuse may be way up. We just don't know about it. Kids with learning disabilities, behavioral health issues, a lot of them need to be back in school. So, and food insecurity, which is skyrocketed. And yes, a lot of schools are providing, free meals. Many times you have to go to the school to get it, or they're not able to deliver it to the home. So we think food insecurity has gone up a lot. So there's all these disparities that this pandemic has increased and shed light on. And I think we have to be aware of the downsides of continuing to have remote learning, particularly our K through 12 school, students. So I think people want to get the schools back in session, do it safely, and there's some guidance that's being developed and has been developed, and we'll also know a lot more in the next few weeks from other states and countries.
Thanks, Dora. There's a few questions in here. I'm going to try to lump them because of time, but a few about vaccine development. So I don't know, Ed or Melissa want to take this, but what do you think is the most likely pathway to a vaccine? There's some questions about glycosylated spike protein being an issue, and as well as will the vaccines, do you think they'll have similar efficacy to some other illnesses? And someone mentions flu and pertussis.
Well, let me give you an example. Measles vaccine gives you lifelong protection, but the flu vaccine does not. And it's an interplay between your immune system and the uniqueness of the virus. And like Melissa was saying, we're still learning a lot about this virus. Now, there are some realities here. Look, what the questioner is asking are all very rational rational queries, but they are just different paths to the same vaccination strategy of exposing an individual to an antigen so that they develop neutralizing antibodies. The big problem that we have, as was mentioned, is the developmental process to make sure your vaccines are not only work, but are safe. It takes a lot of effort, and because you're vaccinating non-infected, normal people, and then waiting to see if they will be protected by the virus. It's been estimated that if there are top 4 antibodies, given the fact that the infectivity, the infection rate, not infectivity, the infection rate of SARS-CoV-2 of the COVID-19 pandemic is reducing significantly, thank God. That's paradoxical in a sense that means you have to have much bigger studies in order to prove the efficacy and safety. And my friends at the NIH are estimating with the decline that there may be a need for 100,000 subjects in order to prove whether or not 3 or 4 vaccines will actually work. And that's a lot of people here. And And that's going to be difficult. The only last thing is people think that you make a vaccine and it should work. Our biggest concern is antibody-dependent enhancement, where paradoxically, it's not because bad manufacturing, but it's because of the biology of the immune system and a virus. We've seen certain vaccines actually make the symptoms worse when a person gets the virus. And so we absolutely don't want that to happen, especially if you're going to immunize hundreds of thousands to millions of people who are healthy, but who might get the virus.
Thanks, Ed. Melissa, I'm just going to add to that question. Another person wrote in asking about vaccines based on the envelope or membrane proteins rather than the spike protein. Do you have any opinion or thought about that?
I think it might be difficult to develop a vaccine based on the envelope protein itself. But I think the majority of the vaccines, the ones that are the front runners right now, are focused on the spike protein and developing immunity specifically to spike protein.
Thanks. Norm, there's a few questions in here about different forms of testing. I don't know if you read through them. I'm going to try to lump them, but they're asking about the differences between antigen and antibody testing, and then IgM versus IgG. And what's your thoughts on the where we're going to end up with testing for exposure and or active virus?
I was gonna say, it's gotten a little bit confusing because some of the early tests weren't that good. The FDA has upped their game on that, so that is positive. But just to say right now, certainly what's called an IgM, and IgM is something that happens in the early part of a disease. So the expectation with a good test is if you're IgM positive, that does mean that any symptoms that you have is due to the virus. And I would even say right now, even if you get IgG and you're symptomatic, I think we can kind of, at this point, make the jump that if you're IgG positive with symptoms, it may be due to COVID, since there's not a lot of respiratory viruses circulating at this point. What's going to start to become confusing is if you are IgG positive next winter, we're not going to actually know whether that means that you are positive because of COVID or because maybe you got COVID now and just have IgG later on. So there's going to be a little bit of confusion, but just to say right now, with those batches of tests, the antibodies are good later in infection. So it's not going to be good in the first week or so, maybe 10 to 14 to longer time out. That's when you're really, the IgM, IgG tests work much, much better. And then we have these molecular and antigen tests that will take the phase in the very, very beginning. So we're hoping that the antibody tests will be great for screening a lot of people to make sure that we have people that are positive. And that may be good news for you, because if that means you could be maybe taking the frontline jobs, the frontline where you're seeing more of the customers or whatever it may be, and the people that don't have that hopefully protective immunity maybe not be there. But again, there's a lot, I know it sounds wishy-washy, but that's what microbiology is all about, is there's not a lot of hard rules with things. So if you like black and white, don't go into microbiology. That's just not a thing you do.
Thanks, Norm. There's a question here about working across communities like Greater Boston community, et cetera, to collaborate over COVID. I'll comment first on what's going on at the Research Institute here, and that is, we've alluded to how we need clinical trials to really get to the point where we know whether a vaccine works or treatment works and whatnot. And our research enterprise is revved up and specifically focused on the COVID trials to help us enroll patients into trials that are going to provide meaningful answers to a lot of the questions we've been asking. And we are doing that in collaboration with the Boston communities, but also communities across the globe, literally, with registries over genetics, for example. Other thoughts on how you guys are collaborating across COVID research communities? Ed, do you have a?
I think it's massive interaction. It would just take too long for us to define all the interactions that are there. I think everybody is really mobilized to help and to contribute. And, I'll just give you one prime example is, as you know, JAX is really part of our responsibility is to look at the mouse resources for the entire United States, North America, and to a certain extent, the rest of the world. And we very early in the game, mobilized our resources to get at this mouse model that was constructed for the SARS, was constructed in 2007. And we did 1,200 IV in vitro fertilizations, which is all done by hand in order to expand enough to give what is right now the request for 10,000 of these mice, over 250 laboratories across the country and the world, because this mouse is very specific for a COVID-19 infection. It takes the human ACE2 protein, moves it into the genome. So now it really is a unique model. It gets the same disease, the same pathology, and it replicates. It's this kind of stuff that we're doing primarily to not only our own research, but really primarily to send out to the rest of the world, is a reflection of the connectivity and cooperativity that's happening right now.
If I could put a quick plug in too, just to say, even with some of the government resources, you look at the CDC, the state and national level have been unbelievably good. I mean, you ask them a question, they are trying to get back to you as fast as possible. I can't say enough about how the CDC has been helpful to every other part here, whether it be, the biotech community or.
Oops, he froze. Oh, there he goes.
You're back. Good. We lost you for a second, but you're back.
Me?
Yeah.
I was putting a plug in for the CDC. I actually was saying that, I can't say enough when it comes to partnerships. Both the state and the national level CDC have been phenomenal. When you ask a question, sometimes quite often takes a little while to get an answer. They are on things, and I've just been thrilled with some of the partnerships with the government. CDC and FDA, I've never seen them be as fast, as responsive as they are right now.
It's a great plug. And Norm, you got a fan club. There's some people writing in here about your class of '84 at Portland High School. Is
Portland
that right?
High School, yeah.
I feel, am I an imposter since I was Portsmouth, which is New Hampshire?
Portsmouth.
It's Portsmouth.
Doug
and I assume Portland.
So I'm close to New Hampshire. I mean, close to Maine. I'm really close.
All right. Well, we misunderstood. We thought you were a local hero. You're close to local hero. Okay. I think we're getting towards the end of the half hour here. Let me see if I can get another question in here. There's a question about the CDC modeling that's kind of long-winded. I'm going to point Dora to read it. It's up near the top, but it's about how we're predicting the death rates and therefore the asymptomatic patient population. So
I see that. Yeah.
So I'll let you read that while we go to another question.
And I think that I'll just answer at a very high level because I'm not going to read it completely and digest it. But I think, there are all these models, and it's been good. I've enjoyed, watching the different models, but I think we also have to keep in mind. I think there was a famous modeler, mathematician, I forget who it was, who once said, "All models are wrong, but some models are helpful." And I think that particularly where this is a novel virus, we've had people, the state's had people, we've had people spend a lot of time modeling it. And they've all been kind of off because they're, I keep saying it's a novel virus. We really don't know how it's going to react. And so you can model it, retrospectively and say, oh, well, here's the current data and see, how it looks on a curve. But I think the main thing I think to keep in mind is that we are way, way far from herd immunity. In fact, the R0, there's some couple of recent studies showing the R0, that is how many people an average person spreads it to, is probably much higher than, it may be much higher than it was thought. It was thought to be, 2.3, 2 to 3, and there's some recent ones, probably you've seen it, 5.7, which is, and actually Augie Valenti is an epidemiologist at Maine Medical Center. He and I go back decades, and we've been through too many outbreaks and pandemics now to speak of, but we both said that early on, it feels like it's more than 2, but and sure enough, it seems like it may be more than that, maybe 5 to 6. if that's the case, then what happens is you need the to attain herd immunity. You need a lot more proportion of the people of the population immune, and maybe in the 80s. So if you think, a lot of vaccines, I mean, we're lucky. We have a good year when the flu vaccine is 75-80% effective. So the fact that, and of course, this particularly hits older people worse, and they tend not to react to vaccines as well and amount a good immune response. So even if you had a vaccine that was, pretty, 80% effective, you're not going to get, 100% of the population to be, vaccinated. And so it's, we are, even with the vaccine, and I seem like I just wonder if we're going to have one in the next year, but I hope so. But I just feel like it's also a little bit wishful thinking, but I don't know as much about the vaccine development as people here on this on a call. So it just feels to me like even when we get a vaccine, we are going to be dealing with this for a while. This is, again, chapter 2 right now of a long book with a lot of chapters, and 5 years from now, we're going to be dealing with this. So to me, no matter what model we're looking at, the biggest challenge we have is changing the culture so that people are masking. These are the tools we have: masking, physical distancing, hand hygiene. We've got to do. Those are the tools we have, and we are at risk for surges, and significant surges, if we're not careful and not do all that. So I'll conclude my remarks with that.
That's a great way to wrap up, is with the advice to all of us. I apologize that we didn't get to all the questions in the panel, but I wonder, Agnieszka, if we can ask our panelists to be available for answering those questions electronically. If people want their question answered, how do people feel about that? Getting an email or getting an email filtered by Agnieszka? Sure. There was one request that came to Dora and or me to get the screening questions that MaineHealth is using. Oh, yeah. And we can do that. We can get that to Agnieszka.
And I should just mention that the product, that app, is called Conversa. It's a company. I think the company that makes it is Conversa, CONVERSA. I should just look it up on my, and they, so you can actually, I think, go to their website and probably get the information. Yeah. But we have to answer this every day when we come into the office. So,
well, thanks everybody for all your input, and we'll give it back to Agnieszka to take us home.
Okay, so it is 4:31, so our time is up, but I just wanted to thank every panelist on this webinar for your time and your super interesting and expertly remarks. I think it was extremely interesting and helpful. I've been getting a lot of emails just congratulating us on the great panel. And a lot of people have been asking if this will be available as a recording. So I'm going to answer that now. Yes, it will be. We probably need a couple days just to make it, available. So probably next week. But thank you again so much. And now I'm going to say, go outside, enjoy the beautiful Maine weather.
Thank you. So nice to see the panelists too.
Yeah, thanks everybody.
Thanks.